Hello, everyone. We're happy to see you today at the Adipose Bionetwork seminar, which is a part of the Human Cell Atlas Biological Networks series. We want to remind you to join the Human Cell Atlas by visiting our homepage or scanning the QR code. You can join any of the 18 biological networks. You can join more than one. Please visit our webpage to email us and indicate your interest. If you would like your publication to be included in the HCA publication list, please visit our webpage and follow the instructions on submitting your publication for the inclusion. Please stay tuned for the upcoming HCA events. We are excited to have the first HCA Middle East Symposium in November 15th, 16th. It's virtual. Visit our webpage to register. The HCA Asia annual meeting is happening this year in Kolkata, India, in the end of November. And we are looking forward to seeing all of you at the HCA Town Hall on December 14th. The registration is upcoming. Today's seminar agenda, we're featuring two speakers, Anne Loft from University of Southern Denmark and Lucas Massier from Karolinska Institutet. For those of you who are logging in live today, we will follow with a discussion. For those who are watching us on YouTube or Bilibili, we will record the talks, but not the discussion. So with this, I would like to introduce our speakers today. So our first speaker, as I mentioned, is Anne Loft. She is an assistant professor at the University of Southern Denmark and a group leader in the Center for Functional Genomics and Tissue Plasticity, ATLAS. Her research is focused on understanding how adipose tissue contributes to the development of obesity-related comorbidities in humans using single-cell sequencing technologies. If you have questions for Anne for her talk, please use Q&A function of Zoom, and we'll be happy to address them. Anne, please go ahead and share your screen, and welcome.
Thank you, Ellen. Do you see it?
We do.
Perfect. Okay. Hi, everybody. So today I will actually show you some brand new data on how the adipose tissue adapts to different modes of weight loss in obese individuals. So I guess we can all agree that the adipose tissue is really a remarkable organ. It has fantastic capacity for storing energy and also for expansion to accommodate a nutrient overload. So when people start to become obese, their adipose tissue expands, and that can happen by different ways. So we have this hyperplastic growth, which is basically just differentiation of pre-adipocytes into mature adipocytes. That is generally regarded as a healthy way of expansion with adequate adipogenesis and little inflammation in the tissue. And then we have this, on the other hand, we have this hypertrophic growth, which is regarded as a more unhealthy way of expansion with little adipogenesis and very much inflammation in the tissue. That then also confers a risk of developing these obesity-related complications. So we and others have recently used single-cell sequencing technologies to investigate how the adipose tissue adapts to diet-induced obesity in mice. So here we just show you the pen. So here we fed mice a high-fat diet for 18 weeks, and we isolated the epididymal white adipose tissue, and we then isolated the nuclei to perform single nuclei RNA sequencing on. And this we did because these conventional single-cell sequencing technologies do not allow us to capture the adipocytes. So we could recover all of the major cell types in the white adipose tissue at We could then zoom in on to the individual cell types, for example, the immune cells, where we could see that the fraction of macrophages really increased in response to high-fat diet feeding in mice. And we could further zoom in on these macrophage, and because the resolution is so good in this data, we could subcluster the data and identify these subpopulation of macrophages. And here we could also study the fractional shift in this compartment in response to high-fat diet. And here we saw that these perivascular adipose tissue resident macrophages, they actually decreased in fraction, whereas these lipid-associated macrophages really increased in response to diet-induced obesity. And we could also look at the transcriptional signatures of these different macrophage subpopulations, where we saw that, for example, the perivascular macrophages, they turned on genes that were linked to lipid handling, really indicating that they required a lamb-like phenotype and likely contributed to macrophage lipid handling or as obesity arose in these mice. So in humans, there has also been some recent studies using single nucleotide sequencing technologies to study what happens in the adipose tissue as obesity arises. And many of the same changes that are seen in mice are also seen in humans. For example, the macrophages, and especially the lipid-associated macrophages, the fraction of these increases, whereas the smooth muscle cells, the endothelial cells, and also these fibroadipogenic progenitor cells, or short FABs, they go down, which likely reflects that the capability of the adipose tissue to perform adipogenesis and angiogenesis decrease in obesity. So what is less understood is what happens in the opposite situation when obese people loses weight again. So it is known that the fat cell shrinks, and it's also known that inflammation goes down, but which type of inflammatory cell that is responsible for these changes is not really so well understood. So we decided to investigate this in much more details using this single nuclease sequencing approach. So for this, we conducted a longitudinal studies where we enrolled obese individuals, so 7 females and 7 males, to a bariatric surgery. And we then took out a biopsy of their subcutaneous adipose tissue, subcutaneous abdominal adipose tissue, both at the time of enrollment, and then again following 5 to 10% weight loss due to a low caloric diet that the participants had to undergo in order to get this bariatric surgery. And then we also followed the patients after the surgery and took out the last biopsies 2 years post-surgery. And this then allows us to study different modes of weight loss. So we have the more short-term moderate weight loss that all of the participants accomplished within a period of 6 to 12 months. And then we have this longer-term bariatric surgery-induced weight loss. Yes, and if we look at how the people lose the weight, it becomes clear that already in response to this moderate or shorter-term period, they lose a significant amount of weight, but of course much more in response to the bariatric surgery. And we also see an improvement of different parameters, for example, linked to insulin sensitivity. And we also recorded their weight between the visits where we took out the biopsies, where we could see that following the surgery, the individuals experienced a gradual weight loss, which peaked at 12 months and was sustained until 24 months or 2 years when we took out the last biopsy. Okay, so we then isolated nuclei from the adipose tissue and sequenced, and then we performed some different clean up of the data to get the high quality nuclei. We also genetically demultiplexed the different nuclei into the individual donors, and we've performed some additional filtering of the low quality nuclei and also some ambient RNA removal, and thereby ended up with 125,000 nuclei in total, which we could then analyze on. And here I have shown you a UMAP of the different cell clusters that we recover from the human adipose tissue, where we see all of the major cell types in the tissue, the adipocytes, the fibroadipogenic progenitor cells, the smooth muscle cells, and endothelial cells, the myeloid and lymphoid immune cells. And all of these different clusters are really covered by a really high number of nuclei. And as expected, we also see a very distinct gene signature in these. So the first question we asked was how these different different cell types in terms of fraction responded to the different modes of weight loss. And here it became apparent that there was not much going on, at least in terms of compositional changes in response to this moderate weight loss. Whereas especially the endothelial cells that went up and the myeloid and lymphoid immune cells went down in response to the bariatric surgery. So we wanted to explore this in further detail for the immune cell compartment, where we digged into the myeloid compartment that we subclustered and recovered different subpopulations of the myeloid cells. So here we identified the adipose tissue resident macrophages, which we further subdivided into more regular ATMs and then antigen presenting ATMs based on their levels of certain antigens. We also identified large clusters of the lipid-associated macrophages and some foam-like macrophages, which resemble the macrophages that has been described in atherosclerosis. And keeping in mind that at least these ATMs and LAMs have been described to be induced in human obesity, we ask how this looked in the opposite direction or situation, sorry. And here we saw again that there were only really minor insignificant changes in response to the moderate weight loss induced by the diet, where we again saw a very dramatic decrease in response to the bariatric surgery. So it really seems that upon this moderate weight loss, where people only loses 5 to 10% of the weight, and therefore, of course, still is quite obese, there's a requirement for the macrophages to be there and to contribute to the cleanup of lipids in the tissue. So we next turned our attention to the FAB subpopulation and subclustered the data and identified 4 different subclusters of FABs. So here we have the DPP4 FABs, which likely represents a multipotent interstitial progenitor cell that, at least in mice, have been shown to contribute to both basal and high-fat diet-induced adipogenesis. And we also identified some PBAR gamma FABs that expresses high levels of adipocyte marker genes and likely represent some more committed pre-adipocytes. We also identified clusters of CXCL14 FABs and EPA3 FABs, where the latter, at least in mice, have been proposed to be some adipogenesis regulating FABs that do not have a high capacity to undergo adipogenesis themselves. So one thing we tried, which was to perform trajectory analysis to map the trajectories between these different FAB subpopulations. So by doing that, we identified 2 main developmental trajectories, and here one branch went from the DPP4 fabs all the way to the adipocytes, and we termed that branch the adipogenesis branch. Then we also identified another branch, which we the no adipogenesis branch, that likely ends up in these EPA3 fabs, supporting the findings from mice that these may be not capable of undergoing adipogenesis, but more of function to regulate the process. And at the moment, we are carrying out RNA velocity analysis to further map the transitions between these different fab subpopulations. So we further explored how the proportion of these fabs is regulated in response to the different modes of weight loss. And here it's known that in human obesity, these different fab subpopulations really trend to go down. And when we look at it in response to weight loss, the most significant change is really the quite strong induction of this DPP4 fab already in response to this moderate weight loss induced by the diet, and this increase in the fraction of these last all the way through the weight loss period induced by the bariatric surgery. So this increase really, to us, suggests that there's a mechanism that rises the fraction of these DPP4 progenitor cells during the early weight loss to really restore normal hypoplastic potential in the adipose tissue, to allow for this early adipose tissue remodeling. So we also evaluated the transcriptional changes in the fab compartment, and here we found more than 1,500 genes that are differentially regulated in one or more of the fab subpopulation in response to the weight loss. And what is most significant is maybe that these DPP4 fabs and PPAR gamma fabs, it really seems that it is here that the most significant transcriptional induction of genes occurs, whereas transcriptional repression of genes is mostly happening in these CXCL14 fabs. And we can also cluster these genes based on their temporal patterns, or not temporal, but based on their response to these different modes of weight loss. And here we see that cluster 1 contains the genes that responds to both moderate and bariatric surgery induced weight loss, and is, for example, genes linked to insulin signaling. Then we have these cluster 2 genes, which contains the genes that is upregulated in response to bariatric surgery, for example, TGF-beta2. And then we have this cluster of downregulated genes that is mostly downregulated upon the bariatric surgery-induced weight loss, which consist of genes linked to apoptotic processes. So we also carried out a functional enrichment analysis and looked for common pathways that were enriched in the different clusters. So, for example, the genes that are induced in response to a moderate weight loss are enriched for pathways related to energy metabolism, so indicating that the fabs become more metabolically active, likely because of their role in this early adipose tissue remodeling. And furthermore, the genes induced by the bariatric surgery, they belong to pathways that are related to migration and extracellular matrix organization, supporting that the fabs really have a very active role in the tissue remodeling also at this time point. And finally, we then, as I already mentioned, see that the genes that are repressed by bariatric surgery belongs to pathways related to apoptotic processes, which of course makes sense since the adipose tissue becomes healthier. So today I have shown you some snapshot of our findings from this study. And we are, of course, analyzing all of the changes that occurs in the different cell populations that we identify. But already now, I think it's very clear that there are some comprehensive compositional and transcriptional changes in response to the different modes of weight loss, and this is highly specific to the individual subpopulations that likely reflect their distant roles in the process of this healthy adipose tissue remodeling. So what I showed you today is, for example, for the macrophages, that the different subpopulations of macrophages, they are not really depleted or reversed by this moderate weight loss, which likely indicate that the macrophage lipid handling is really important in these early phases of the weight loss. And then I also showed that this this Fab compartment, these DPP4 Fabs becomes the fraction of these are increased already in response to the moderate weight loss, which likely indicates that the adipose tissue already at that point is restoring its hyperplastic potential and its ability to undergo adipogenesis. So by that, I want to end by thanking all of the people involved in this project, all of my great colleagues in Atlas, and also the clinicians at Sydvestjysk Sygehus. And thanks to you for listening.
And thanks so much. It was a great talk. And I see we have a question already. From Katie Whitetalk. Great talk. And I was just wondering what statistical test was used to determine differences in cell type composition between groups?
So we used a Kruskal-Wallis test, I think it's called, if it makes sense.
Thank you. The next question is from Ibrahim Al-Zaim. Thank you for a very interesting talk. I was wondering if you see an augmentation in endothelial nuclei at visits 2 and 3, and if you try to subcluster the endothelial cell population.
So we actually see induction in the endothelial compartment in response to the bariatric surgery. And it makes sense, I think, because, as I said, so the endothelial cells are also depleted as obesity occurs. So I think that the fact that we see a restoration in the fraction of the endothelial cells at that time point, it reflects that the adipose tissue has its capacity to perform angiogenesis and become healthier at that point.
Thank you. And probably last question for you for now from Ulrich Stiefel. Thank you for this nice talk. And have you looked at mature adipocyte populations? And is there a cluster which only appears after weight loss?
We have looked at the adipocytes. I would say it's not clear. We see some polarization of the different adipocyte clusters that also responds especially to the bariatric surgery. But I would not say that there's an increase in the fraction of one of the adipocyte subpopulation in response to bariatric surgery. I think that I can say.
Thanks, Anne. There are more questions in the Q&A section. If you have time, you can type the answers, or we can try and address them after the talks during discussion if there's time. But thank you for the questions. And with the
Ellen, you muted yourself.
I muted myself. Okay, sorry about that. So we're happy to go to welcome our next presenter, Lucas Massier. He's a researcher at the Karolinska and the Department of Medicine and a team leader at the endocrine unit, focusing on adipose tissue heterogeneity using single cell methods. Recently, Lucas has been focusing on single cell and spatial transcriptomic studies of human adipose tissue with a specific interest in adipocyte subtypes, fibroblast heterogeneity, and adipose tissue microarchitecture. Lucas, welcome and thank you. Please go ahead and share your slides.
You see the slides?
Yes, we do. Thank you.
Okay. Hi, everyone. First of all, thanks for the invitation to present our thoughts on the comparability and integration of white adipose tissue single cell data here today. In the next 20 minutes, I'd like to briefly talk about the difficulties of white adipose tissue single cell analysis. I will go talk about some difference between comparing and integrating this data. I will briefly mention the currently used integration methods. I'd like to focus a bit on adipocyte heterogeneity. And I also briefly compare human and mouse datasets. And most of the slides you will see today are from our recent publication on this specific topic. So just closing this here because it's blocking my screen. We think that white adipose tissue is a very challenging tissue for single cell analysis. And this is because, for one, there are various adipose tissue locations, and we know all of them have or can have distinct functions. Here's an example showing 15 of these adipose tissue locations, and this includes various subcutaneous depots, various visceral adipose tissue depots, but also depots such as epicardial or perirenal adipose tissue. And if we look at the transcriptomic data of these various adipose tissue depots, we see that there's quite a difference in the transcripts that are expressed. Of course, mainly split into the visceral and subcutaneous depots, but we also see quite drastic changes within, for example, subcutaneous depots of various locations. And these differences might, of course, be explainable by differences in cell type populations. Now, there have been excellent studies also on the single nucleus and single cell level, comparing, for example, subcutaneous and visceral adipose tissue. And we are also currently working on depot differences, focusing more on different visceral adipose tissues, namely omental, mesocolic, epiploic, and mesenteric adipose tissue. And this is just as a sneak preview because today's topic is something else, but we indeed see quite drastic differences in the cell type distribution, mainly because between subcutaneous and other visceral depots, but also between or within the visceral depots, as you can see here in this heat map. Adipose tissue is also not very well defined and not very organized. This is from a publication where we performed spatial transcriptomics on adipose tissue. And we see we have some small regions where we have like vessel structures or something that we can recognize. But overall, we don't feel that there is an organized structure behind the distribution of the cell types or spot transcript information. And this is in contrast to, for example, brain tissue, where when we use spatial transcriptomics, we can recapture quite well a ground truth that we know from histology, for example, of these layer structures. But we also see that if we would just use an unsupervised clustering or just using the marker genes, we will not get to the same kind of layers that we actually know of. So here, semi-supervised clustering methods are helping. So for adipose tissue, it might be the case that we are just lacking the ground truth because we don't know how it is organized yet, so that we can choose or develop better clustering algorithms. Furthermore, adipocytes specifically are very hard to work with. And this figure is taken from Margaret's presentation, but paper. But adipocytes are so big that we cannot sort them and do a drop sequencing on them. So we have to collagenase digest the tissue and take the fraction that was like the stroma vascular fraction, so without the adipocytes, or we perform single nuclei sequencing so that we can also capture the adipocyte information. But we also know that both of these methods have inherent differences that are important to consider when analyzing this data. And for example, this is shown here, and this is nothing to do with adipocytes, but this is a publication on retina tissue of rabbits. But here, Santiago et al. Compared on the same sample single nuclear and single cell sequencing extensively. For example, they saw differences in the abundance of protein coding transcripts, but also long non-coding mitochondria and transcription factor transcripts. And it was quite an extensive number of genes that were differentially captured by the specific methods and that also play a role in very specific biological pathways. So this is something that one should consider when analyzing one or the other method. Now we try to integrate or compare the previously published single-cell data sets that are available for adipose tissue. So we included various data sets of subcutaneous adipose tissue, omental adipose tissue, and perivascular white adipose tissue, covering a big range of age range, but also range of BMI. And then the first things that we thought of, how can we analyze this data? So we can either compare it by just analyzing each data set separately, or we can try to integrate all the information and then do a combined analysis. And yeah, so here, first of all, we wanted to compare the data. What we did is we analyzed all the data sets individual using the same pre-processing step and clustering algorithms, etc. And we calculated like a similarity index between the cluster marker genes that we identified. And we see that all these data sets are actually quite similar with one exception. And then using these clusterings, we performed this kind of guided network analysis. And each dot here represents one cluster of an individual study. And we see when just by comparing the marker genes, these clusters segregate quite nicely into groups of myeloid and lymphoid cells here in blue, and endothelial cells in green, adipocytes in purple, and the fibroblasts and mesothelial cells in orange. So that tells us that the data sets are quite similar, and we thought that the integration would be feasible. But we also see differences, for example, in the abundance of specific cell groups. For example, we see higher abundance of vascular cells compared to in single nuclei sequencing compared to single cell sequencing. This might be leading to an overrepresentation of the vascular cells in single nuclei sequencing data sets. Yeah, so with this, I'd like to make a small excursion to integration methods, and there's a ton of integration methods, and there's new integration methods being published every year. And there's also a number of very good studies comparing all these integration methods, and I just have 3 examples here on the left side. And in this one example, Chen et al. Discussed various pre-processing steps, normalization steps like SC transform or log CPMs. And they also talk about the data integration. And I think all the 3 examples that they make here can be applied to adipose tissue. We can have a case of very similar biological samples if we compare different individuals in one, like clinically, in one cohort with the same clinical background. We can also have shared subpopulations between sample. For example, if we look between different subcutaneous depots, which are quite similar, but not completely, or if we compare inflamed compared to healthy adipose tissue, where we know that there's an influx of macrophages or other immune cells. But we can also have quite distinct cell types between samples, for example, when comparing omental to subcutaneous adipose tissue, where we know that we don't find mesothelial cells. Now, in this paper, they only compared a couple or a handful of integration methods, but there are many more, as Tran et al. Tested here in this paper. However, we think that Harmony and SCVI are currently the most employed integration methods, and we tested them alongside BPKNN and the surrogate method RPCA, and we find that they work well for adipose tissue data, and we assess the integration efficiency here by the KPET method and the adjusted RUND index. And I just wanted to mention that in the new surrogate pipeline, both these methods, Harmony and SCVI, are also now included, which of course facilitates the analysis of new datasets and also the comparison of different integration methods. We also see that after integration, we still retain differences between single nuclei sequencing and single cell sequencing, which is what we expect because of the inherent differences. And I think if we would see like a 50-50 distribution here, would mean that we would have over-integrated our data, which is always also a point that we want to avoid. But this is, of course, hard to judge. I just want to give one example here for the endothelial cells. Here's again another score that can be used to assess integration, local inverse Simpson index. And we see in this UMAP that both methods here colored in green and purple are integrated fairly well. And then in this combined UMAP, we can, of course, annotate our clusters, and we see that marker genes are nicely overlapping with marker genes reported in other studies. And specific cell types are in the integrated data sets nicely located to specific clusters. And we see that for marker genes, for example, for lymph endothelial cells, pericytes, endothelial cells, and also vascular smooth muscle cells. And this integration then allows us to do some basic depot comparisons. And we see, for example, that the cluster of lymph endothelial cells, cluster 6 in orange, and also these kind of endothelial cells here in pink or purple or what, are upregulated in omental and perivascular adipose tissue. And we confirmed this by deconvolution of bulk sequencing data in independent datasets. Contrary, we did not find that we could integrate adipocyte clusters very well. And here exemplarily, I showed two UMAPs colored by cohorts and depots. And we see that there is not a good integration. In general, we could say that we saw very good integration of vascular and immune cells, which were very conserved across cohorts. A little bit less for fibroblasts, but we saw the least integration for adipocytes, at least for the single nuclei and single nuclei data. We proceeded to look at this a bit more systematically. And of course, if we analyze individual data sets, we do see different adipocyte clusters. Here in Evans' data and in our data set here, we see different adipocyte clusters, and adipocytes make up about the same proportion of cells or nuclei. However, we saw that only very few marker genes were overlapping between these adipocyte clusters. And then if we looked at the overall fold change of marker genes within adipocyte clusters, we observed that they are quite low compared to the background information, or much lower than, for example, compared to vascular or immune cell marker genes, which suggests to us that they are less specific. We then did another comparison where we compared between the cohorts how many of the marker genes between the clusters are overlapping. And we saw for immune cells and vascular cells that there was a high overlap of marker genes, meaning that they are very similar and very conserved clusters. But for adipocytes, we saw less of an overlap of marker genes. We also compared the single nuclei sequencing data, specifically of the adipocytes, to bulk sequencing data of isolated adipocytes, and also to the adipocyte sequencing information from our spatial transcriptomics cohort. And we found that the bulk isolated adipocyte data and spatial are more similar compared to the single nuclei data. We then wanted to explore if we can find out which genes might be responsible for these differences. And here we took the data from the Phantom consortium, which is cage data of isolated cell types throughout the whole body. And we looked for adipocyte-specific genes. So here in green are genes that have a fold change that is very high compared to all the other cell types included in the Phantom consortia. So these seem to be very adipocyte-specific genes based on the cage sequencing. And then we looked at their distribution in the spatial sequencing versus the single nuclear sequencing. And we see that some of these genes are highly expressed in the spatial, but not at all in the single nuclear data, while others are highly expressed in the single nuclear data, but not so much in the spatial data. And these genes are shown here. So we see the two clusters, the genes that are high in the spatial data and also in the bulk data, and then the clusters or the genes that are high in the single nuclei data sets. And then we overlap that with the marker genes that we used in our spatial paper to identify adipocyte subtypes. And interestingly, some of these genes or most of these genes overlap with the list of genes that are highly different between the single nuclei and the spatial data, and also the bulk data of isolated adipocytes. Now, why that is the case, we are not sure, but I think that we think that this might be a reason or an explanation why we maybe see less heterogeneity or like less systematic heterogeneity of adipocyte clusters in the single nuclei data. If we look at our spatial data, however, so in our spatial data, we reported the presence of 3 adipocyte subtypes, which we called AdipoSAA, AdipoPlin, and AdipoLab. And we also did some protein validation of them. And we currently have a new spatial data set, which was, so the subjects are collected in Germany. So it's a completely different center and it's different persons handling. And we also have an improved bioinformatic pipeline. However, we still see the same adipocyte clusters. I mean, the expression of marker genes is a bit different, but we can recapture these adipocyte subtypes. And also in a recent publication by Wittek et al., they did full-length transcriptomics on adipocyte, single-cell full-length transcriptomics on adipose tissue. And if we take a look on the adipocyte subpopulations specifically, we see that they also can recapture a leptin-enriched subtype and a plin-enriched subtypes. So we think that there is adipocyte heterogeneity, probably on a reduced level compared to other cell type groups. But like there's differences in the methods used, and these will affect which kind of adipocyte subclusters we can capture. And so this is my last slide, just to compare the human clusters to the mouse data, and it was just done for the sake of this talk. So there's no fancy like figures, but similar networks, as I showed before, just comparing marker genes in a similarity index between the human datasets and the mouse datasets. And we see that there's a high overlap for vascular cells and immune cells. I'm zooming in here, for example, for the mouse B cells in purple and the human B cells in orange. So high concurrence between the human and mouse data. Same, for example, for the T cells. And we see also a high overlap between the fibroblasts in mouse and humans. In contrast, we again see a lack of overlap for the adipocyte sickness, which may, of course, be because we are not even sure in the human single nuclei data what we should learn or take away from the adipocyte subtypes. With that, I'd like to end my talk, and I hope I could convince you that due to depot heterogeneity and the loose organization and adipocyte size, white adipose tissue is a challenging tissue for single-cell analysis. that the integration of wide adipose tissue single-cell data sets is feasible due to an overall high similarity between the individual studies. However, integration methods need to be chosen carefully and will always introduce a new bias. Adipocyte clusters lack reproducible heterogeneity or cell type specific clusters. And except for the adipocytes, human and mouse data aligns fairly well. With that, I'd like to thank you for your attention, and I'm happy to answer any questions.
Lucas, thanks a lot. We have quite a few questions. I'll try to get through maybe 3 of them today. So the first one is from Boxiang Liu. Nice work. Do you firstly integrate all the data sets to classify 4 clusters? If so, what algorithms do you use firstly and for that step?
No, so we did analyze each data set separately. We annotated the clusters to reference, like we manually annotated all the clusters based on literature work. And then we took out the cell groups that I presented, so vascular cells, adipocytes, fibroblasts, and immune cells, and then only integrated these specific cell types, because then the reason, and that's also like discussed in the literature, because then the background is more similar, which facilitates or improves integration. And we also compared that to the SC-ANVI tool, which takes prior clustering information into account for integration, but it was very similar result.
Thanks. Are cell nuclei doublets a major concern for adipose tissue?
Can you repeat the? I didn't.
Are cell and nuclei doublets a major concern for adipose tissue?
I don't think they are any more concerned than in any other tissue. We try to remove the doublets by the available bioinformatic tools. Yeah, but I don't think it's specifically or worse for adipose tissue.
Okay, and did you have to remove mitochondrial and ribosomal genes prior to integration of different modalities, single nucleus versus single cell, or was that not necessary?
We usually exclude these genes in our pipelines.
Okay, great. If you have time, please go ahead and answer the rest of the questions in writing, if you want.
Yes.
So,
I need to stop.
yes, thank you. And, oh. Can you see my screen? Yeah. So with this, I just wanted to thank those of you who are joining us, who are viewing us on YouTube or Bilibili. If you would like to join the Adipose Bio Network, please either email us or scan the QR code and indicate your interest. And with this, we're going to stop the recording.